Hello everyone, I am presenting my research work in 23rd International Symposium of Control Release Society, and the title of my research work is Innovative Dry Powder Inhaler Device for Enhanced Pulmonary Delivery in Animal Model. Now dry powder inhaler markets are rapidly growing market, and various kind of the disease condition like asthma, COPD, lung cancer and tuberculosis. This delivery is choice for the treatment. These are the few marketed product available. Now dry powder inhaler is a dynamic field of pharmaceutical and research. And here for preclinical study, the dry powder delivery device are necessary and these kind of the devices are not readily available. In preclinical dry powder delivery devices, there are two kind of the devices, whole body exposure and nose only exposure devices. Now in whole body exposure devices in which the drug are dispersed in a chamber which leads to the non-specific absorption of the drug to the various other part of the body. At the same time, the dose requirement is very high. While in case of nose only type of the devices, the absorption is very precise at the nasal route. At the same time, the effectively we can deliver the drug to the targeted site, which minimize the waste. Now, various kind of the nose only devices are reported in a literature as well as available in a market. The first kind of the traditional devices has the major concern is uncontrollable airflow, while in case of second type of the nose only devices has the issue of uh, clogging of the devices and cleaning. Now third kind of the device are demand the complex handling. At the same time, the fourth type of the devices has the issue of inconsistent deposition in a lung site, at the same time breathing variability. Despite of these advantages, the challenges are persist in achieving the uniform and efficient pulmonary delivery for the animal. So the strong need is recognized to develop a novel type of the devices which can overcome the disadvantage of existing devices. At the same time, these devices are simple, easily fabricated and cost effective, and it can be easily used at the screening stage. These are the various components of novel fabricated devices, and this is the diagrammatic features. The key advantage of this device is the aeration is supplied from the bottom and which will give the laminar and Brownian movement and which ensure the uniform dosing and prevent the turbulence in flow. At the same time, the mouthpiece and spacer is provided, so which prevent the choking of a nose, which is safer for the rodent. To validate the fabricated devices, here we are selected the rifampicin and ofloxacin drug, and these drugs are suitable and useful particularly in multi-drug resistant cases of tuberculosis, and these are the benefit or advantages associated with the pulmonary delivery of rifampicin and isoniazid microparticles. Now in research lab we have successfully formulated the ofloxacin and rifampicin microparticles by spray drying technology. These are the operating conditions for the spray drying process. The formulation were optimized and characterized. The particle size, entrapment efficiency, and PDI value found to be excellent. And these formulations are suitable for the in vitro validation of novel devices. And this research work has been published in the Journal of Current Drug Delivery, Bantam publication. Now, in order to validate the novel pulmonary device, the ofloxacin and rifampicin particles are fluidized with a different time duration and with a different dose. And the recovery of that drug is found to be 40% in case of ofloxacin, while 51% in case of rifampicin. And these studies are demonstrated that The recovery of the drug is or depend upon how much drug is loaded and time duration. Now in vivo validation of devices performed in male Vista rate. Total 4 groups are evaluated: IV, oral, inhalable, and control group. At the end of the treatment, the blood, bowel fluid, and lung homogeneity were analyzed by the HPLC method. These are the precipitation methods are used for the extraction of the drug from the blood, plasma, and lung homogenated, and these are the process condition for the HPLC. Now, these devices effectively deliver the drug in a deeper part of the lung and which can be reflected by the higher percentage of drug delivery in a bowel fluid and lung homogeneity. Now, in vivo drug deposition study of ofloxacin and rifampicin are revealed that there is a higher deposition of the drug is found into the lung homogeneate, while in case of the ratio of lung to plasma ratio is comparable, and ofloxacin has a higher drug retention as compared to rifampicin. So based on experimentation, we can conclude that the fabricated device is a cost-effective and efficient tool to assessing the performance of this formulation for preclinical study. And these devices are reliable platform to evaluating the efficiency of the formulation. Thank you.